Dear Nobel Laureates, Academy member, teachers and students of Solna Gymnasium, it's a pleasure and a great honor for me to welcome you all to Solna and to this webinar. My name is Jonas Egervärn and I'm the headmaster here at Solna Gymnasium. We are of course sorry that you couldn't be here in person, but still very excited to meet you here at Zoom instead and get this unique chance to take part of your research and scientific findings. Your results are at the cutting edge of research and will surely be part of the solution for future and current problems. As you probably all agree on, especially these days, progress of science in general feels more important than ever. I also would like to welcome you to Solna Gymnasium. Solna Gymnasium is just 5 minutes north of the center of Stockholm city. It's an upper secondary school educating young students since 1948. Here in Solna, we have the advantage of being in a very dynamic and research-intense region, which enables for our students to have a close cooperation with higher education, such as our neighbor Karolinska Institutet. The science institution here at Solna Gymnasium have a long tradition with a high standard, with motivated students and competent teachers. And it is inspiring for us to meet Professor Doudna and Professor Charpentier here today, and perhaps get the possibility to hear how it all started for you. And I would not be surprised if we have future laureates among our students. So once again, to all of you, a warm welcome. And please let me now introduce and welcome Professor Claes Gustafsson from the Royal Swedish Academy of Science.
Thank you very much. And I'm very pleased to be here and to meet all the students, even if I don't see you just now, but I will share with you my screen actually and show you some just a couple of slides that I've made just to tell you a little bit about the Nobel Prize and what we will hear today. I will not talk about the subject, but a little bit of how we work in to award Nobel Prizes. So the Nobel Prize in Chemistry, where did it all start? Well, it started with Alfred Nobel's will. So in 1895, Alfred Nobel wrote a will, and in his will, he stated that he wanted to donate his money and that it should go into prizes in science and in peace and in literature. And if we look a little bit more closely at that will, there are some passages that are especially relevant for the Nobel Prize in Chemistry. And one of the things that Alfred wrote was that the prize in chemistry should go, and all these prizes should go to those who during the preceding year have conferred the greatest benefit to humankind. And the last part of this sentence is still valid. We still try to sort of identify individuals, scientists that have made a contribution that has conferred the greatest benefit to humankind. This with the preceding year is something that we cannot really do because it takes a long time for us to evaluate different prize fields and it also takes time for scientists after they discover something to publish and to sort of gain general acceptance and so on. So this is something that we have changed. And the prize in chemistry should go to the person who made the most important chemical discovery or improvement. And this is actually a little bit different between the different prizes. So the medicine prize can only go to a discovery. They don't have improvements as a possibility. And in physics, they have something called innovation. So their innovations are actually a possibility to give prizes to. So it differs a little bit between the prizes. And you might think that this is just wording, but it's actually something that we take seriously. So when the medicine prize is awarded, they think hard if this is a discovery or if it's an improvement. It's something that's important for them when they decide. And it cannot only be innovation, and neither for us. It cannot only be an innovation, it needs to be something more. And we also need to not give any consideration to nationality, but the prize should be awarded to the worthiest person. So this is very important, that where you come from, your gender, your religion, or whatever, that should not matter. The only thing that matters is what you have contributed with and what you have discovered. This is the way that Alfred wanted it. So he was, one can say, before his time in many ways. So how does it work? Well, this is the, I'm sorry for the resolution, which is a little bit bad, but this is a typical year in the Nobel Prize Committee and how we work. So in September, we sent out forms for nomination where we ask institutions, former Nobel laureates, we have a large number of specially invited scientists and so on, and all professors in chemistry within the Nordic countries. They get invitations to nominate for next year's Nobel Prize. And somewhere in the beginning of February, there is a deadline for submission, and then we get this pack of nominations that we start to go through. And if someone is nominated more than once over the last 10 years, and it's not by the same person, we will evaluate that person. So we evaluate basically all the nominations that we get. More or less all of them are evaluated in some sort of way. And then we start evaluation work. So we read papers, we send out to external experts, we have internal experts, and we go through these nominations and we try to understand what it's all about and see what has been discovered and who has done what. And then we start writing reports, and we do that in June or August somewhere. And in the beginning of September, we go to the academy and we say, we have what we think is a winner here. And then if they feel the same, then it's taken up to a vote, and that's in early October. And directly after the vote, we call up the laureates, and sometimes that can be in the middle of the night, depending on where you live in the world. So if you're an American, this can be sort of a traumatic experience. We will call you around 2 or 3 o'clock in the morning. And then we have the Nobel Prize award ceremony in December. And just to give you a feeling of the sort of volumes here. So we send out about 3,500 invitations every year to nominate. We get 600 answers, so 600 different nominations, and we have about 400 different nominated candidates every year. Everyone is evaluated in some way. And we write about 50 written reports, and they are both from external, mostly from external, but also from internal experts. And we have months of meeting discussing this. And for many of the prizes, I say all the prizes, they have been with us for many years. So we do this over and over and over again. And often before we award a prize, we have evaluated both 10 and 15 times, and sometimes even more than that. Some prizes can take up to 25 years before we can award them. So there is a process. And then it all ends with the Nobel Prize Award ceremony and banquet, and that's December 10 every year in Stockholm. And that's because that was the day that Alfred Nobel died in Sanremo, Italy in 1896. And the flowers that you see, that's always so beautiful at this Nobel Prize award ceremony, they were actually from the city of Sanremo, who sort of donate them every year in the memory of the great inventor. And this year, we are extremely happy to have 2 Nobel Prize laureates that has been awarded the Nobel Prize in chemistry for the development of a method for genome editing. And it's Professor Jennifer Doudna. And Jennifer is a professor at University of California, Berkeley in the US. And it's Emmanuelle Charpentier, who is the director of the Max Planck Unit for the Science of Pathogens in Berlin, Germany. And with that, I'm done with my introduction, and I'll hand over the floor and the word to Emmanuelle Charpentier. So please, Professor Charpentier.
Okay. Thank you very much. So now I'm talking, I guess. Hello, Jennifer. Hello, Jennifer. I will start. It's okay with you? So, dear students, so I guess Jennifer and I, we are very happy to discuss a bit of science. At least me, I will focus on the science. We can always ask also general questions later about the career of both of us, if you are interested in how it works when one wants to become a scientist. So I will start, and I'm sure that Jennifer after will anyway, I have to share my screen, will anyway complement my talk with additional information. In general, we are well complementary when we give our talk, so then this is good. So I have now to go back and share the screen. Yes. Okay, so I guess now you should see the screen. So actually, Jennifer and I were awarded the Nobel Prize in Chemistry of this year for the development of a method for genome editing. So I will not be surprised that if you were telling me that you know a little bit about this technology. So in principle, it's a technology that allows to edit the genomes of cells and organisms. So it's called CRISPR-Cas9. And on the slide, you can see the target DNA that can be modified by the tool. And you can see scissors that are actually representing the protein Cas9. This is a protein that has the ability to recognize the target DNA in a sequence-specific manner, thanks to an RNA component. And this protein can cleave the DNA, so at a very specific site. And this protein can be programmed with an RNA molecule that will allow to recognize a specific site on the target DNA. And this RNA molecule, you see it drawn inside the scissors. And in nature, this is composed of the so-called CRISPR RNA and tracer RNA. So, and for the purpose of using the technologies, those two RNAs were linked together with a linker loop. I'm going briefly to tell you, because I think at least our time are quite limited, to tell you how the project started. This is, I guess, what is important to convey to you. This is really basic science. This technology is used as large applications in the field of life sciences. I will come back after to the applications very briefly, but I guess what is the message that is important to provide you is that this technology, programmable scissors to modify DNA of cells and organisms and recode in principle life, it is important that you understand that it's really basic science. And I will start by telling you that the project started by focusing on a bacterium called Streptococcus pyogenes, and that is a strict human pathogen that causes a large range of diseases. So this bacterium can be found only in the human host. So some human beings are colonized by this bacterium without causing diseases, but you have some clinical isolates that can cause severe diseases. And like any organism, specifically like any bacteria or any macroorganism, Streptococcus pyogenes encounters a number of stresses. So as a matter of fact, for Streptococcus pyogenes in the human host. So the bacterium has to cope with a lot of environmental conditions, and the bacteria have to survive in order to cause diseases. And you have different regulatory networks that are entering into play and that are actually, how do you say, made possible thanks to molecules such as proteins, also molecules of RNA in nature, also the types of very small molecules, chemicals that work together and allow the bacteria to produce virulence factors when it is about to cause diseases, or to produce different kind of factors that contribute to the fitness of the bacteria and the fact that they will be able to adapt and survive in their environment. So we know that a certain type of immune system, I guess you're familiar with it with regard to the SARS-CoV-2 infections, which we are currently experiencing. We have an immune system that allows us to fight infections by bacteria and viruses. And actually, bacteria can also be infected by viruses, and it's really part of their environment. They will encounter a number of so-called mobile genetic elements. This can be bacteriophages, so the viruses of bacteria. This can be also plasmids, so ectopic pieces of DNA that have the ability to replicate on their own, and that in the case of Streptococcus pyogenes, for example, viruses having the ability to lyse the bacteria, so really kill the bacteria, or they have also the ability to bring to the bacteria new genes that will contribute to the fitness of the bacteria and their ability to actually cause diseases. So there is a complex interaction between bacteria and mobile genetic elements. And the same occurs between archaea and mobile genetic elements. You have also viruses that can be infected by viruses. And like any organism, bacteria have evolved to be able to cope with these invading genomes. And they have evolved diverse immune systems that are classified mainly as innate immune systems. And around more than 30 years ago, there was the first discovery of elements of a system that was found later on to be an adaptive immune system existing in bacteria and archaea, allowing them to defend themselves against attacks by viruses. And this is what we call the CRISPR-Cas system. So in principle, the way it works is as follows. When a phage infects a bacterium, it will inject its own DNA, its own genomic material. And this will be recognized by the CRISPR system that is composed of a series of genes, the CRISPR-associated genes, Cas genes, that code for proteins, the CRISPR-associated proteins. And in addition to the CRISPR-associated genes on the genome of the bacteria, you have also the so-called CRISPR array that is composed, and here you see it with black sequences, composed of very short repeats. So in the sequence of the CRISPR array, you have these small repeats that can be actually of maybe 3-4 repeats to more than hundreds of repeats, and that are interspaced by unique sequences that have a specific origin. Those sequences are actually sequences matching mobile genetic element sequences. And this is actually reflecting the number of mobile genetic elements which the bacteria will have encountered. So in principle, and this is thanks to this so-called step of the immune system that is spacer acquisition, when the invading DNA of the phage is recognized by the CRISPR-Cas machinery, there will be a portion of the DNA of the phage that will insert into the genome of the bacteria at the level of the CRISPR array. So here, for example, it will be a green sequence of the phage. And this will trace in a way the history of all the mobile genetic elements which the bacteria have encountered. And this will work as a memory device, so that upon a second infection, the bacteria have memorized the genome through acquisition of a part of its genome, and then we'll be able to bring the machinery into place and recognize the phage upon a second infection. And this is thanks to this CRISPR array that is transcribed into a long CRISPR RNA molecule that will undergo processing, cleavage, to lead to short CRISPR RNAs that each are composed of this specific sequence of one specific mobile genetic element. And the mature CRISPR RNAs will associate with a complex of CRISPR-associated proteins and will therefore guide the complex of CRISPR-associated proteins to the cognate phage. So there will be recognition of the phage through the memorized sequence of the phage at the level of the CRISPR RNA and the cognate sequence on the genome of the phage. There will be base pair interaction between the RNA and the DNA. And one of the CRISPR-associated protein will recognize by this base pairing system the specific sequence on the genome of the phage, will cleave the genome of the phage, and then it is the dead end for the phage, because cleavage of the DNA is not mostly not repaired in bacteria, and so there is no replication that can proceed, and the phage DNA cannot replicate, the phage particles cannot be produced and propagate, so this is the dead end for the phage. So this is how the system is working. Now, this system has largely evolved in the sense that this composition of CRISPR-associated genes, for example, and the nature of the repeats of the arrays have largely evolved in bacteria and archaea. So you have a large diversity of CRISPR-Cas systems that have been identified. There is also a large history of the CRISPR-Cas, and you can ask questions maybe after the talk of Jennifer and I, but you have a number of researchers that have identified those elements, worked on other types than the system Jennifer and I focused on. But in principle, there is a large evolution of the CRISPR-Cas systems with small, let's say, divergences with regard to how the system is working for, really the precise way the phage is recognized and the precise way the machinery is brought into place, and the precise way either the DNA or the RNA of the phage will be recognized. So it's a fascinating research, but in principle, we focused on one system in Streptococcus pyogenes that was really unique, because in this system, in addition to the CRISPR-associated genes and the CRISPR array, we found another RNA molecule that is called tracer RNA. So the system, instead of working with a complex of CRISPR-associated proteins and the CRISPR RNA, works essentially with only one CRISPR-associated protein and two RNAs. So to make a very long story short, using a genetic approach and a biochemical approach, the story is as such. The system works with two RNAs that form a duplex that undergo processing, meaning the RNAs need to be activated through maturation and through cleavage, and leading to this type of machinery where you have the Cas9 protein guided by a duplex of RNA, and that can cleave site-specifically the DNA. So this was simplified further by simplifying this duplex of RNA by linking the two RNAs together to lead to a single guide RNA. And very fast, we understood that in principle, this would provide a lot of advantages to the biologists who had access to a large number of genetic tools. For your knowledge, molecular biology is a field that is 50, 60 years old. And a lot of genetic tools have been developed over the last 50, 60 years. That's why biologists have been able to clone DNA, to amplify DNA, to sequence DNA, to also modify DNA, to insert a piece of DNA into the genomes, to delete a piece of DNA from the genomes, and so on and so forth. But there was always a little bit, sometimes a tool that would not be appropriate to be delivered to a certain organism or a certain cell, or too complex to use for certain organisms or certain cells, or not precise enough, or really cumbersome. And this is what the CRISPR-Cas9 brings compared to the other technologies. It's easy to program, it works very efficiently, and it has been shown to very, very early on to be able to be delivered in actually using the delivery tools that were available. And for sure, the last 8 years, since Jennifer and I, we published our paper in Science in 2012, you have more delivery systems that have been developed. So I'm sure Jennifer will comment on this. In principle, the technology can correct DNA, delete a piece of DNA, add new DNA, replace a gene or a sequence of DNA in a very precise manner. And it has been shown to be working very fast and very efficiently to perform modifications on genomes in plants, in model organisms such as flies, fishes, or mice, in organoids, and in human cells. So biologists have been quite happy with the technology. And the reason why it's transformative, actually, and it's recognized as one of the most transformative technologies, is that with this technology, actually, we figured out that indeed the biologists were in need of a powerful technology that would allow really more precise genetics. And so it has been very useful in research and development. We are always, as biologists, interested in understanding the functions of genes and understanding the mechanisms of life, whether we are interested in plant biology or in pure fundamental research working on model organisms, or whether we are interested in clinical research. To do research, we need tools, and to discover new mechanisms, we need a large panel of technologies, including genetic technologies, and CRISPR-Cas9 really makes the difference. It really accelerates the way we can generate transgenic animals, or the way we can understand the functions of genes in human cells, the way also we can find, for example, in the biomedical research, novel targets for novel therapeutics, where we can develop models of diseases that allow to test therapeutics in development. So this is, for example, in human medicine. In plant biology, it also allows to anticipate let's say, the resistance that the plants develop against novel challenges of climate. So it allows to accelerate also the production of crops, but also to produce crops that are safer. And you have also a direct application of the technology in medicine, because it's developed as a gene medicine combined with cell therapy to treat certain human genetic disorders, or like blood disorders, but also used in cancer immunotherapy. And you start to see some patients that are effectively cured by CRISPR, specifically patients who have blood disorders. So this is where I will end. Just a last slide, because I like to put these slides for acknowledgements. Science is about collaboration. This story was a collaboration with Jennifer Doudna, as you know. From my side, it has involved a number of funding agencies and research institutions, and the work was actually developed from my side, in my brain at least. I mean, during my time at Umeå University, that was from 2008 to 2013. So it's a Swedish story from my side. So I'm very happy to let you know that Sweden and Umeå was very good for me to develop this project, and also the very nice collaboration with Jennifer Doudna, who will take over, I guess. Jennifer, I have to stop sharing my screen. Okay.
Thank you very much, Dr. Charpentier, for a very nice presentation. And we will not take questions now, but we will just hand over to Dr. Doudna, who will continue the presentation. Thank you. So please.
Very good. Thank you very much. And it's a great pleasure to be here, of course, with my colleague Emmanuelle, who just gave a lovely introduction to the CRISPR system. And I will sort of take it from where she left off and talk a bit about where the technology is now going. So let me get this going. Okay, there we go. Great. So yeah, so CRISPR, as you heard from Emmanuelle, is a technology for changing DNA and for doing it in a precise way that allows scientists to make targeted alterations to the code of life. And so I'm going to break my talk into three parts. I'll just say a very little bit about CRISPR, since you already heard this lovely talk from Emmanuelle that described the origins of the system in bacteria and our work together on how it works. And then I'll tell you a bit about genome editing and where that is headed. And at the very end, I will say just briefly a few words about how CRISPR is being used right now in the face of the coronavirus pandemic as a detection system. So as you heard from Emmanuelle, CRISPR is a technology for changing the code of life. And the way it works is by targeted cutting of DNA. And so this is a video that actually shows the CRISPR-Cas9 protein with its RNA attached to a piece of DNA, and right there you saw the DNA get cut. And that's fundamentally what CRISPR does. And in bacteria, this is a great way for bacteria to defend themselves against viruses. So here you're looking at this is a scanning electron micrograph of the surface of a bacterial cell that is being infected by viruses. And so if this cell has a CRISPR system, it can grab pieces of DNA from the virus, store them in the bacterial genome itself as a little record of the infection, and then make pieces of RNA from that encoded viral genetic material and use those little pieces of RNA to find the virus if it shows up in the cell and cut it like you saw in that little video. And so in nature, this is a system that is very effective. So this is showing a cartoon of some bacteria growing somewhere, let's say in the soil. And here's one getting infected by a bacteriophage. And so the CRISPR system allows storing a piece of the viral DNA in the bacterial genome in this place called the CRISPR that has a pattern of sequences, as Emmanuelle explained. And so the cell can then make a copy of those sequences in the form of RNA. And then those RNA molecules are cut up into units that each have a sequence that comes from a virus. And these RNAs then combine with a second type of RNA called tracer and a protein called Cas9. And together, that RNA-guided protein is able to search the cell looking for matching sequences. Sequences that match the sequence in the RNA. When that match occurs, the DNA unwinds, the protein cuts the DNA. And in bacteria, those double-stranded DNA cuts cause destruction of the viral DNA. So it's a fabulous way for bacteria to fight a virus by literally using the virus's information against it, using its own genetic information to destroy it. And in the research that, that my lab did with the lab of Emmanuelle Charpentier and two fabulous scientists who led that work in each of our labs, it was Martin Jinek in my lab at Berkeley and Krzysztof Chailinski in Emmanuelle's lab at the time in Vienna. And these two scientists were able to figure out not only how Cas9 cuts DNA, like you saw, but also how it could be harnessed to do something different in other types of cells. Because we realized that although in bacteria, double-stranded DNA cuts lead to rapid DNA destruction, in eukaryotic cells, that means in animal cells or plant cells, certainly, or human cells, a double-stranded cut to the DNA triggers DNA repair. And this was not our research. This was based on a large body of research that had been done over the 2 or 3 decades before we did our work on CRISPR. And so a lot of scientists had realized that when plant or animal or human cells experience a double-stranded break to the DNA, they detect the break and repair it. And that DNA repair process involves correcting the broken DNA by, in some cases, making a small change to the DNA sequence during the repair process, or in other cases, integrating a new sequence of genetic material at the site of the original break. And so earlier technologies for genome engineering relied on this principle of introducing a double-stranded break to DNA and then allowing the cell to repair it and make an edit to the DNA at that precise position of the repair. And the challenge to those earlier technologies, as Emmanuelle implied, is that they were difficult to manipulate. They were hard to use. You had to design proteins that would recognize a specific place in the DNA sequence of a cell and cut it. And those technologies could work, and they had everybody excited about the principle of genome engineering, but they were difficult enough to use that a lot of scientists and a lot of laboratories were not able to really, take advantage of that technology. And this is where CRISPR comes in, because who knew until the research that was done to understand this bacterial immune system, that in nature, bacteria had already figured out how to make targeted cuts to DNA. And not only that, how to program an enzyme, Cas9, to make cuts wherever you want. And that's really fundamentally what our research was about, was figuring out that process of DNA cutting, how to control it, and then how to apply it as a genome editing tool. And I want to show you one more video that just illustrates how we imagine Cas9 working in a eukaryotic cell, where the DNA is inside the nucleus, and Cas9 with its guide RNA, when it goes into a cell like this, where you put it into a cell, we imagine that it can search the genome looking for a sequence that matches the sequence of the guide RNA. And this is, by the way, based on actual crystal structures of Cas9 solved in our lab, showing that Cas9 forms an RNA-DNA duplex, leading to DNA cutting at a precise position. And then that broken DNA is handed off to repair enzymes that can fix the break by introducing a small change, like in this example, or in some cases, integrating a whole new sequence of genetic material at the site of the break to insert, for example, a whole new gene into the genome, but to do it precisely, do it exactly where a scientist programs it to happen. And that's what's really awesome and exciting about CRISPR, is that it really is a precise technology that, as Emmanuelle said, is relatively easy to manipulate, and it's relatively easy to use in any cell type. And so it meant that all of a sudden, scientists all over the world had an opportunity to study biology using genetic manipulation in a way that was really never before possible. And so so that sort of takes us to, where the technology is headed. And again, as Emmanuelle said, this is a technology that is truly cross-cutting across biology, because it allows scientists to do experiments to ask fundamental questions about the function of genes in all sorts of organisms, like studying how the genes in a butterfly control the wing pattern and color pattern that we see on their wings. And that's actually been done now using CRISPR, for example. It's also being used in medicine, and I'll say a bit more about that in a moment, in ways that help us not only understand the genes that cause disease, but to actually correct disease-causing mutations at their source, which is incredible. And then also in agriculture, using CRISPR to make changes to plants at the level of their DNA that could do things like increase the nutritional value of plants, make them less susceptible to drought, and make them more, more resistant to various kinds of diseases. And all of these things are incredibly important, especially as we globally face the challenges of climate change. So just to drill down on one specific example, I think that, we've been really, it's been just extraordinary to see over the last 8 years, the development of CRISPR as a technology to cure genetic disease. And this is a great example that Emmanuelle alluded to for curing one of the most sort of best characterized, best known genetic diseases called sickle cell anemia, or sickle cell disease. And this is a disease that results from a single base pair mutation in one gene of human cells. Imagine that. It's a single base pair that is kind of miscoded, miswritten in the DNA. And in patients that have 2 copies of this gene with this mutation in it, their cells make a form of hemoglobin, which is the protein that carries oxygen in our blood, that is susceptible to aggregation. And that means that their cells form this classical sickled shape that leads to clogging of their blood vessels and organ damage and just terrible pain in their life. And they go through kind of repeated crises where they have to be hospitalized in many cases. And amazingly, so CRISPR is a technology that can go in and literally change this mutation, correct it back to the correct mutation, or the correct sequence in the gene, or it can actually be used to turn on the production of another protein called fetal hemoglobin that normally is turned off when we're born. But in these people, in these patients, CRISPR can be used to turn it back on, and it will substitute for this mutated form of adult hemoglobin. And that's exactly what's been done for a number of patients now. And this is work that has been led by CRISPR Therapeutics and their academic partners. And this is a company that Emmanuelle and her team started. And this is a patient, Victoria Gray, who's actually been treated and effectively cured of her sickle cell disease using CRISPR-Cas9, using the work that we published together in 2012. And I can tell you as a scientist, it's unbelievable to see. I mean, it's just, it really cuts to your heart to see that your work has actually impacted somebody's life like this and, really improved their life. And now it's not just Victoria Gray, it's now a number of patients that have been studied over many months. And it looks like the CRISPR technology is working very well for not only sickle cell disease, but other types of genetic diseases of the blood. And we think in the near future, it will be useful for a lot of other kinds of genetic diseases that you might have heard of, like cystic fibrosis and muscular dystrophy, and potentially a lot of other things as well. So it's very exciting times for sure. This all needs to be done with some care because, of course, the CRISPR technology is a very powerful tool, as you can see. And so I've been involved in particular over the last few years with the kind of global discussions around the ethics of CRISPR-Cas9. This is a report that was released fairly recently from the national academies that talks about the really kind of evaluated the technology, and in particular, the way it might be used in the future in human embryos to make heritable changes to DNA that don't affect just one individual, but actually also can be passed on to future generations. A really profound sort of use of CRISPR that I think needs obviously very, very careful thought before proceeding. This report establishes criteria that we certainly hope will be adopted globally by scientists that might be doing that type of research. And then I'm just going to end by with one slide about the coronavirus. And again, this circles back to right at the end of Emmanuelle's talk. She mentioned that one of the interesting biological properties of CRISPR is that it's very diverse in nature. There are many different types of CRISPR-Cas proteins and lots of variations on this theme of RNA-guided DNA, or in some cases, RNA targeting. And that's exactly what's been harnessed for the purpose of using CRISPR as a diagnostic. And I'll just show this very briefly, but in work that I and some colleagues of mine in the Bay Area published very recently, we've established a rapid test for the coronavirus, the virus that causes COVID-19, using a form of CRISPR that can detect the viral RNA and report on it very quickly. And it's just on the right-hand side, this is the principle of how this works. So we use a CRISPR protein that has a guide RNA that can interact with the RNA of the coronavirus. It's an RNA virus. And when that interaction occurs, this CRISPR enzyme is activated as a little cutting machine, and it will cut pieces of RNA that have dye molecules appended to them. And when these RNAs get cut, the dye molecules are released and they fluoresce and they give a very nice signal. And so this is a great way to detect any kind of RNA, but it's very useful for detecting the coronavirus RNA. And we're hoping that in the near future, this will be a test that we can actually use in real settings, like on our Berkeley campus, for example, to help us detect the spread of the virus before it can cause disease. With that, I want to just conclude and point out that this is a really exciting time. We're still understanding the fundamentals of the technology. We're also trying to figure out how to deliver these CRISPR-Cas molecules into different kinds of cells and tissues and control the kinds of edits that get made. And of course, this all needs to be done with an eye towards safety and effectiveness, as well as how people around the world can get access to this technology by making sure that it's affordable. And I'll just stop there and invite you to learn more if you'd like to at innovativegenomics.org, which is a website for the institute that we started in the Bay Area that focuses especially on these aspects of ethics and affordable access to genome editing. Thank you.
Thank you very much for a very nice presentation. And it's a fascinating story going from basic scientific studies of a bacterium all the way up to sort of diagnosis of viruses and treatment of patients. And you should all remember that this is in it's only 8 years ago that the first sort of papers were published. So it's been an extremely rapid development, and I'm sure it will continue that way for the next 10 years. So with that, I'll just leave the word to Niklas Martos, who will take care of the Q&A session and moderate that.
Thank you. Thank you so much for the presentations. Can everybody hear me?
Yes.
Great. Well, my name is Niklas Martos. I teach maths and chemistry here at the Solna Gymnasium. And I think that's enough of a presentation about me. So if these students would like to present themselves.
Hello, my name is Selina, and I'm in the 3rd year of the Natural Science program here at Solna Gymnasium.
Hi, my name is Filippa, and I'm also a 3rd year student of Solna Gymnasium.
I'll try break Lamin if you would like to introduce yourself.
Hello, my name is Lamin, and I am also a student at Solna Gymnasium in the third year.
We're just waiting a second for Omar to load his screen, I think.
Hi, my name is Omar, and I am a student at Solna Gymnasium.
So I'd like to start off by giving the word to Selina, who has a question about the professor's origins of how they got into science, I think.
Yes. Hello, Professor Charpentier and Professor Doudna. So our first question to you is, have you always been interested in the natural science subjects, and especially chemistry? And when you were in our age, were you eminent students, or what did you think about school?
So maybe Jennifer can start, because she can bring some exotism to the subject because of where she was living when she was younger.
I don't know. I think Paris is maybe more exotic, Emmanuelle, but sure. But I'm happy to start, and thank you, Selina, for that question. It's really a pleasure and an honor for us to be here with you guys today. So I grew up in Hawaii, and I grew up in a small town, and I went to public school. Nobody in my family was a scientist, but I always liked math, and I actually had a wonderful chemistry teacher in high school that taught us kids that, you know, science is a process of discovery. It's about figuring things out, you know, doing experiments to find out things that nobody knows right now. And I thought that was really cool. And we had when I was probably in the same, it's the same stage of school that you guys are, we had a program in the state of Hawaii that brought scientists from around the state to talk about their research. And we had, in one of the talks that I remember very well, we had volcanologists, we had marine biologists, we had astronomers, I mean, people doing really interesting stuff. But the one I remember was a cancer biologist who came in and talked about her research to try to understand the chemistry of cancer cells. Why do normal cells become cancerous and start to grow out of control? What's the chemistry of that? And I have to tell you, when I heard that talk, I thought, that's what I want to do. That's exactly what I want to do. I want to do that. I want to be a biochemist. I didn't really know what it was, but it looked really fun and really interesting. And that's really always been kind of my pursuit. And, you know, afterwards, you know, in college and later I always was kind of trying to, you know, find ways to pursue my interest in biochemistry and work on problems that I thought were really interesting.
Yeah, so from my side, as we discussed prior to this session, actually, you know, in France, you have the system, obviously, like in Sweden, that is that starting at 15 years old, you have 3 years of school, whereby you already have to choose your specialization, at least the last 2 years of the 3 years. And so this is at 15 years old. So I needed to choose whether I would like to do sciences or literature. And I chose biology and mathematics. So this was then to be at 16, 17, 17, 18, at première D and terminal D. D, it's D, and it's the baccalaureate D, and D, it's biology and mathematics. Actually, for me, it must have started already when I was 11, 12 years old. I remember very well my professor of biology, and there was also a professor of physics and chemistry when I was 14 years old. At least from physics and chemistry, what I remember is that we were doing some, for sure, very easy experiments at the bench, and not dangerous experiments, but some experiments at the bench. And I think I understood as well that being a scientist, at least in those fields of chemistry, biology, would imply also a work performing experiments. And I guess most likely I like the idea of a process whereby you do experiments and you have to find out something. But at least what my mother told me when later on I chose to do my master thesis at a university at the Pasteur Institute, so working on bacteriology. When I later knew the lab that I had chosen, she told me, that when I was 11, 12 years old, I came back from school and I said, one day I will work at the Pasteur Institute. So most likely this was the influence of my teacher in biology at the time. I was not in Hawaii, but I was in a very small, in a kind of suburban city, 25 kilometers south of Paris. So not very exciting actually, even though we could go to Paris. But yeah, I just got interested in science by, obviously very much by school. And my father was working for being responsible of gardens and parks in a city. So he was very close to nature, and he would, I would hear him telling a lot the names, the Latin names of plants. And my mother was working in the medical sector. So maybe this has also influenced me in a way to continue and do life sciences. Mm-hmm.
Thank you so much for your answers. Going in a bit of a different direction, Omar, you had some questions about using the CRISPR-Cas9 technology with regards to bacteria. Yeah, would you like to elaborate the question? We're having some technical difficulties, so Omar is coming to my computer to enter the screen.
I'm meeting Paul. There is a growing problem with multi-drug resistant bacteria, where our antibiotics are becoming less effective with time. Could CRISPR play a role in solving this problem by eradicating the resistant gene?
So maybe I can start answering. So actually CRISPR originates from bacteria and archaea. So one would think that one could harness the system for technologies in bacteria. And as a matter of fact, this has been started. I'm not so sure whether it will lead to a kind of new antibiotic that will be commercialized in the market. But in principle, this is the ability to use CRISPR technology, to use the CRISPR technology and to repurpose it so that it can be delivered to a bacterium that one would like to kill. With the idea of, a CRISPR system that works as an antibiotic. And in this case, the CRISPR technology will be targeted to recognize specifically the genome of this bacterial cell or a specific bacterial strain. And what happens in bacteria when you cleave DNA is that the repair systems are overall very inefficient. So you will end up affecting the replication of the genome, and this will be the dead end for the bacteria. The issue with this technology is more how to deliver the CRISPR technology to the bacteria. And this, it's the same challenge that one wants to deliver an antibiotic, but in this case, one has to deliver the CRISPR system really inside the bacteria. And this is the issue of having a system that can reach the right tissues and cells in which the bacteria are, let's say, surviving within the human host. So there is a biotech company, at least one biotech company that is working on this repurposing of the technology to work as an antibiotic and kill bacteria, and potentially be an alternative of the antibiotics that are conventional antibiotics used in nowadays.
Thank you. Professor Doudna, do you have something you would like to add, or do you feel like your colleague has answered the question?
I think she summarized it very well.
In that case, I'd like for Lamin to ask a question that might be a bit on the darker side.
Okay, so can you hear me?
Well, I can hear you at least.
Yeah, I can hear you.
Yeah.
Okay, nice. This question is mainly directed towards Professor Doudna. The question is the following. Could it be dangerous allowing such a powerful method of genetic modification into the possession of individuals who could weaponize it by, for instance, creating genetically modified soldiers?
Well, the short answer is yes, but I would just point out that, you know, I think with any powerful technology, there's a risk of misuse or even intentional, you know, misuse, and CRISPR is no different, you know, and this is you know, it's one of those interesting situations where the reason that CRISPR is so transformative as a technology is that it is relatively easy to get a hold of and to use in different cell types. And the flip side of that is that you know, it means that it could be potentially misused or used to cause harm. And I think that's one of the challenges that we face in the field going forward is how do we make sure that it continues to advance, and it, you know, it's, I mean, it, you know, the whole field is moving forward at a very, very rapid pace right now. And, you know, there are very exciting opportunities on the horizon for CRISPR, but I think we all want to make sure that, you know, that the outcomes are positive. And so I've been really an advocate for transparency, meaning open discussion of what's happening and getting scientists around the world to be involved in those conversations. And, you know, and I think that I've been, you know, cautiously, I'm optimistic that, you know, there's been a really good partnership globally among scientists right now to put together guidelines that will really, you know, be the criteria that people will use going forward with CRISPR. And that being said, you know, just to your specific example of super soldiers, one of the things that makes that also not really feasible right now, probably thankfully, is that you know, that those kinds of manipulations would require many genes to be changed, and we don't know which ones would need to be changed. And so I think there's a lot that would need to be done to understand the genetics of complex properties of human beings that, you know, is still probably many decades ahead. So that's kind of just on the practical side. But I think ethically, it's super important that people are already kind of thinking about this and grappling with the challenges of a powerful tool that has great potential to do good, but also has the potential to cause harm.
Thank you, Professor. Professor Charpentier, would you like to weigh in on your thoughts?
Yeah, I just would like to add that that in any case, I believe it's unlikely that the technology comes to a point that in any case you can target more than one gene, or at least genes necessary to be targeted for human transformation in general or human enhancement. But that I also wish to see the technology to not be used for human enhancement in the future. So yes, that is my personal opinion, and that the technology, at least for the use of the technology in humans, it's really to cure diseases, and I believe it should be the focus should be on really this application in humans.
Thank you. Philip, would you like to ask your question that you had about the future?
So now that you have worked with CRISPR and won the Nobel Prize in chemistry, what do you see as your next step? If you would keep working on CRISPR, then what with CRISPR are you trying to develop? If not, then what are you looking to do? Is there maybe another project you have your eyes on?
Thank you. I suggest that Professor Doudna starts, and then I might have to interrupt, but give the word over to Charpentier since we're a bit low on time.
Yeah, I'll be very brief. Well, in my lab, I'm continuing to work on CRISPR biology and uncovering some of the newer mechanisms that are still being detected for CRISPR, and fundamental questions about what these CRISPR systems are doing in bacteria. I think is still very interesting, and lots of things still to be uncovered there. So part of my lab works on those kinds of projects, but a growing proportion of my group and actually at the institute that I'm at, we are working hard on applications of CRISPR. And as I mentioned, with a focus on CRISPR affordability. So it's really, really exciting to see companies moving ahead with CRISPR therapeutics, but the challenge is going to be cost, because those therapeutics are very, very expensive. And so when you think about how people around the world will get access to the technology, we're going to need to continue to do things that will bring down the cost and make it more accessible. And that's what a number of us are now focused on, is ways to do that. And one quick example is in the example of sickle cell disease that I mentioned. Suppose that a day arrives when we can actually deliver the genome editing into a patient without a bone marrow transplant, that we had a delivery vehicle that would just target the editing molecules directly to the blood cells that need editing, and do it in a kind of a one and done treatment. So that's the kind of thing that we envision, and we're working to develop the tools to do it.
Yes, so me briefly, I mean, personally, I hope to still have time to do research. But having said this, we still work on CRISPR, but the main line of the research of my laboratory, I will say forever, has been to understand how bacteria cause diseases in humans, and to a certain extent how the humans react to bacterial infection. So it's a lot of macrobiology, molecular biology, bacterial, let's say, I should say, working on innate immune systems, and see, their roles in being able to fight bacterial diseases. So it's a little bit my line of research. So we work on different types of mechanisms that allow to gain more knowledge. And my ultimate goal, I would say, will be to find a new mechanism, but that may not lead to a gene technologies this time, but more to an anti-infective strategy. But I'm not sure whether I will get there with my team. Mm-hmm.
I think we all hope you do. Thank you for your answers. I think we have time for one more question. So Celine, if you would like to ask one.
Yes, so can you please describe the day you succeeded, or the day you made your discovery, and how did it feel and how did it happen?
Yes, so first, it's very important to mention that it's a long-term process. It's not something that happens from the sky. So one is prepared in a way. I mean, let's say, we hope we are always prepared for the unexpected. Unfortunately, in science, what happens often is that we don't see always, let's say, the results going in the right direction. But sometimes it happens. So in the case of the CRISPR project, it was mainly first, this discovery of this additional RNA molecule, and actually relatively simple experiments that were to show that there was a need for an additional component at some point on the CRISPR pathway, and to show that you know, the CRISPR-Cas system was working with a protein together with 2 RNAs cleaving the DNA, and then the simplification of the system. Having said this, as Jennifer mentioned, you have scientists at the bench doing the experiments, and they are the first being excited because they are the first, you know, seeing the results. They are, you know, designing the, you know, the experiments, the cooking recipe in a way. You know, they follow the protocol of the recipe and they see the cake, you know, coming up. And so it was an excitement for all of us, but it starts first with their excitement that transmit after their excitement to us, saying, yeah, the experiments have worked. And so these are unique moments. And indeed, you have a lot of moments, and you have those where you know that it's, you know, the key step, you know, so yeah. And this is something that is difficult to express because it's very, you know, it's something you wait for a long time, and when it comes, you know, you cannot believe it, and then you say, but no, I believe it, and so it's a lot of excitement.
Thank you so much. Professor Doudna, if you'd like to answer the same question.
Yeah, I'll be brief, but I agree with Emmanuelle that, you know, science is you know, there are many steps to the process, but I do remember in this case a wonderful day in the lab when Martin Jinek, who was in close collaboration and discussions with Chris Chylinski and Emmanuelle's group, and he ran into my office and he said, you know, Jennifer, we can cut DNA with Cas9 and we can control the cutting. And we literally, both of us ran down the hall to the lab, and we were, you know, leaning over a light box in the lab and looking at, you know, what was really a very simple experiment in a way, you know, and you guys have maybe done this in your labs at school, where you can cleave DNA with a restriction enzyme, and you get little fragments of DNA and you can separate them on a simple kind of agarose gel system. And this is the experiment that he did. And what was exciting was that we could control the DNA cutting by our knowledge of how Cas9 works with its guide RNA. And I think I ran back to the office and immediately, you know, emailed Emmanuelle and, you know, said, oh, this is, you know, really exciting. And as she said, you know, Chris Chylinski was doing similar experiments. And so, you know, there was kind of that wonderful kind of moment when all of the work kind of came together and we realized that this was a really interesting and really potentially really powerful system.
Thank you. Well, first off, as Jonas perhaps heard, this might be doable in school, so maybe we could get some kits. But with that, a great big thank you to both professors, the students, and I give the word back to headmaster Jonas Egervärn.
Thank you, Mr. Martos. Selina, Omar, Philip, and Lamin, good work. Finally, I would like to thank all of you joining us here today. Of course, especially I would like to thank Professor Doudna and Professor and Director Charpentier for taking your time to meet up with us. We appreciate it. And in case you get the chance to travel to Sweden, we would be very happy to welcome both of you here at our school, perhaps next year then. But until then, we hope you do enjoy your well-deserved prize. And thank you, and goodbye from Solna.
Thank you very much. Thank you, and good luck to all of you.